So as usual, we'll have our Q&A session here for the next half hour or so. Again, just as a reminder, this Q&A session is also being live streamed and recorded and distributed. So just an overview schedule for tomorrow. We just had our introduction to antimony network modeling. The 2 sessions in the morning tomorrow will be diving deeper into how to use that to couple network models to cell behaviors, so how to actually realize these in more full CC3D models. We'll again have our hackathon prep session. We'll have more presentations tomorrow. So if you're still not sure which group you want to join, there may be a couple of presentations tomorrow that'll pique your interest. And if not, then we can help you with suggestions for hackathon grouping towards the end of the day tomorrow. Again, we'll have breakout sessions tomorrow in our 3rd session. So after lunch, there will be 2 breakout sessions. Details about that will go out in the morning email in the morning. So make sure you keep an eye out for that. So, and we'll, before lunch, we'll also post those Zoom links just in case anybody misses the email. But information for how to do that and how to join the groups will be provided in the email in the morning. And then we'll follow that up with our first look at compartmental cells and compartmental cell division. So we get into our discussion of, I believe, or no, link starts the next day. But compartmental cells will start tomorrow in our 4th session again with Julio. Just reference our people again, seen the slide 100 times. Any questions? I think is that all the announcements, James, that you can think of? I don't think we had anything else much more than that.
If people had questions that they didn't get fully answered or wanted to bring back up now that we didn't have time to do during the main session, that's fine. Now's a good time to bring them up.
Or if you have questions about your hackathon project as well. I know somebody emailed me earlier saying they might be here for questions for that. So that's also fine to bring up here.
Well, in the case of this neighbor avoiding movement of cells, how different is this from swarming? Like there are no collisions.
So this is, I guess I need to understand what you mean by neighbor avoidance and what circumstances.
Well, when I look at the pictures, either images from the lab or in this simulation,
right,
it looks like the cell is moving between some islands and never touching them, which may mean that the time scale of, maybe they're all moving, but the time scale of that cell is swimming intelligently fast between those semi-static islands.
So I guess I misunderstood the question, because I thought you meant specifically in the case of the self-generating gradient. So that self-generating gradient is a mechanism that's used in things like neural crest cell migration and in lateral line development and in vascular development. And one of its features is that because the cell creates a gradient where the concentration is bigger away from the cell, if another cell gets close to it, it sees the concentration going down, and so cells repel each other. And so you tend to get co-aligned movement of cells. And the simulation that Pedro showed at the very end of the collective migration of the cells, it was a result of that. So in that case, the cells were adhering to each other. So when they touched, they stuck, but they moved collectively as a flock. And so one of the things that you have in classic flock modeling, which we were talking about, is the emergent property of interacting motile agents. If you look at a classic Boyd's model, the agents have to be able to see their environment. They have to be able to say, I want to be given distance from my neighbors. So they have to be able to measure that somehow. They have to be able to say, I want my direction to be the same as my neighbor. So they have to be able to measure the direction of the heading somehow. If you're a bird and you have eyes, then those things are possible. But cells don't have the ability to make those kind of measurements. They can't measure the velocity of something that's at a distance. They can measure stress at their surface, so they can measure shear stresses if they have cilia. But in general, and cells can send out philopodia to touch their environment, so they can explore their environment up to a certain degree. But in general, cells can't see at a distance. They can't see. They can touch, they can smell, they can feel, but they can't see. And so there's no long range signaling except that's mediated by materials. And in this case, what you find is that you get co-alignment of velocity because of the nature of the chemical field that the cells are generating and responding to. The cells can't move over each other because they have a volume constraint. Depending on whether the cells want to stick to each other or not, you'll get different dynamics. And so I think how cells navigate their environment is going to depend a lot on the particular mechanisms that they're using, whether those are mediated by stress in the environment, by density gradients in the environment, either chemical, diffusible chemical, or mechanical. And so I'm not sure there's a universal answer to the general question. I was thinking specifically of the self-generated gradient, where if you create two cells that in that self-generated gradient simulation, you'll see that they repel each other. They'll move to, if they're coming together like this, they'll move and go move parallel.
Yeah.
Well, they won't move away. They'll actually move parallel to each other.
Okay, but they at least don't really collide.
Yeah, but they won't overlap.
So,
they won't cross. They won't cross.
So if I wanted to manipulate that or to exploit that, should I induce some chemical or, I mean, it's now becoming very exciting because the more we understand about these processes, it gives ideas for next step or what utility.
I agree. I think this question about emergence of collective behavior is fascinating. I think simulations like CompuCell or other agent-based modeling methods like NetLogo are very good for exploring that. You get a lot of counterintuitive collective behavior.
So now, what about that situation where lambda changes temporal spatial, like allowing lambda to vary, okay,
Yeah.
as even a complicated function, not being fixed at different values.
Well, so you mean, I'm not sure. So are you asking the technical question about how do you implement variable lambda, or are you asking a question about what controls lambda in biology?
No, I ask a question if the model can take in account a situation like that. And again, if we can induce this kind of variation of lambda to advantage.
Well, certainly adaptation is one of the key things that biological systems do. And we skipped. Pedro had to skip this exercise of the chemotaxis and linear gradient versus chemotaxis in an exponential gradient, chemotaxis with adaptation and chemotaxis without. But in fact, if you don't have adaptation, things fail. If you ask what are the ranges of concentration over which cells respond, the answer is multiple orders of magnitude. And if you have a fixed linear sensitivity, you can't do that. Either you don't move at all when the concentrations are small, or you move or you saturate when concentrations are large. So you have to have an adaptation. And there's some beautiful mathematics and biophysics that's been done maybe 25, 30 years ago, showing that in many cases, cells do exhibit a perfect adaptation, which is what we're modeling here, where the lambda is automatically essentially adjusted to be 1 over the concentration. That gives you the maximum dynamic range of response. Now, in reality, adjusting lambda, for the cell to adjust lambda, it has to change the number of receptors on its surface. It has to change aspects of the signaling cascade inside the cell. It has to change its cytoscalar architecture. So it doesn't happen instantaneously, but it does happen. Perhaps the most dramatic example of adaptation in us is because structurally it's so heavy and expensive, is dark adaptation. When you go from a dark room to a light room to a dark room, your eyes actually synthesize a photoreceptor to make your cells, your rod cells more sensitive. And so they actually are synthesizing photoreceptor. It's a very inefficient way of doing things, but it's also maybe the only way it could happen. It's a good example of evolution picking a good enough strategy and not an optimal one. You'd never design it that way from scratch if you were designing something. But the reason our dark night adaptation takes time is because our photoreceptor is actually synthesizing the photoreceptor. It takes a long time to do that. And it breaks down faster, but when you go out into the sun, when you've been in the dark and you're dazzled, your photoreceptors actually have to break down photoreceptor to adapt. And that's over 6 orders of magnitude, 7 orders of magnitude intensity. Your eye still can see. So that's the classic example of a huge dynamic range that you couldn't get without adaptation. So changing your lambda is a simple surrogate for that. But in chemotaxis, it's pretty good. Pretty good.
Yeah, it was just curiosity, not criticism.
No. I mean, I think it's a great question, and adaptation is really important. And the key there is understanding what the mechanism is. In other words, if you have a model of what the regulatory system of adaptation is, then it's fast. If you don't know, then there are a lot of possibilities. So there was a question about from Louise. Are you still here?
Yeah, I'm here.
So tell me a little bit more about what you're interested in. I think we're interested in the same thing, and my answer is I'm not good at it, but I'd like to hear more about what you think.
Well, I've been doing a number of projects. So one of them's been working on kidney disease, and I've analysed a lot of microarray data sets where podocytes have been subjected to damaging and kind of helpful treatments. And then I've subset a lot of genes that are implicated in damaging effects and healthy effects. And I essentially want to use those matrices of gene expression values to calculate the rate laws and then generate an anemone string based on that. But there's quite a lot of them there. There's about 300 genes. So it's going to take a long time to do it manually. So I was wondering if there's a parser or anything that could be used to kind of automate that process.
Okay, so there are two things there. One of which is sort of a difficult one, And I mean, it's all difficult, but there are two things there. One of which is that you can't get a rate law from expression data from a microarray or a RNA-seq because it only gives you amounts. It doesn't give you dynamic.
Yeah, but it gives you the relationship between the genes themselves.
Well, when you do modulation, then you can at least try to infer network connectivity. And then there are some simple rule of thumbs that you can start out with. Depends on what kind of networks they are. If they're gene regulatory networks or if they're signaling networks or mixes. There is so typically what you get out of the kind of experiments you're talking about are causal relationships. This upregulates this downregulates this. These two things are correlated. If you're lucky, you go to a database like KEGG, and they have some of the pathways already mapped for you.
Yeah, I mean, I've done all of that. But what I'm trying to establish is this subset of genes. I know that they're involved in a particular process that regulates the shape of the podocytes.
Mm-hmm.
So I'm trying to kind of think of a way to make a model that can use the gene expression data. So I know that in damaging conditions, this gene is switched down, so there's only 2 copies of it, but in like good conditions, there's 30 copies, and I can use that information to kind of generate a model.
Okay, so the answer is that the people that we're going to have speaking on Thursday and Friday, the toxicologists, do a lot of that kind of model derivation. I think that they still do it mostly by hand. I've heard talks, the people doing network inference and pathway mapping, of course, are a huge, much bigger world than those of us doing multicellular modeling. They have budgets of 1,000 times bigger than 10,000 times bigger than we do. And their community is about 10,000 times bigger than us. So they're in many ways much more advanced than we are. I don't know. There are a lot of people who claim to be able to do automated versions of what you're talking about. I heard a talk from a group at Harvard recently, whose names I cannot think of at the moment, that had some nice software that did it. There was a group at Penn. The group that I would say probably has, that I've heard recently that had the most elaborate software to do that was from Seattle, was Shmulevich's group. They have that Allen Institute doing systems biology. And they have some software tools that in principle can go from differential gene expression data and at least try to constrain the pathways. I've never used them. I would love to have somebody in my group who is an expert on that. But I typically either build those networks by hand painfully or get somebody else to do it for me. I'm not the expert. I think it's really hard. But I would look at the Seattle Systems Biology Institute and their software and see whether there's anything that they have that you could use.
I'll do that. There's one other thing I wanted to ask you. During the lectures earlier, and it was showing I think it was the amoeba, and it looked kind of similar to the way the podocytes function. Do you know of any models that I could potentially use to modify and make it look like a podocyte?
Well, I think I actually showed podocytes as I thought the most frightening cell type that exists in the body.
Yes.
Because I think that they have a greater structural complexity than any other cell in the body.
Yeah, I've got a model that I've drawn out of the podocytes, how I think they work, and I just want to try and get that kind of running as a proper model, as it were. But I wanted to do like a cellular model of them.
Right. So you want to actually model the patterning that gives rise to the interdigitation of the podocytes. So you want to model the cytoskeletal.
Yeah, essentially. So I've got like some sketches that I've done of how I think it works, and I want to try and test that. by creating a model.
So that's so you might talk to Giulio, who gave 2 lectures on Monday, and it's going to give a few more lectures. He's worked on extracellular matrix modeling and cytoskeletal modeling with explicit fibers, not with CompuCell, with a different package that he uses.
Right, that would
and-
probably cover it then.
And that might be interesting. TJ, who's here today, is writing and will be very soon releasing a software package that can do those kind of fiber models. But the problem there is that when the moment you come to macromolecular assembly, whether it's of the structural proteins that give rise to podocyte architecture, which of course are not as well understood as microtubule, or actin, or intermediate filaments. The problem with fibers is that they have aspect ratio. They're narrow in one direction, long in another. And if you want to know what's happening at their tips, you have to be modeling on the scale of the tip, which is small. If you want to know how they transmit forces, you have to model on the length scale of the fiber, which is large. And so fiber models intrinsically tend to be very computationally expensive. They're beautiful models by people like Alec McGilder at NYU, who would do explicit fibers. But they'll be doing the amount of computation it takes for us to model 100,000 cells over a year. They'll take to model 1 cell microtubule architecture over a minute. That's the kind of computational ratio of effort you're talking about. And so it's not that you can't model subcellular structures to some extent with CompuCell, but macromolecular machinery is intrinsically hard to model. And so then the question is, if really what you want to know is how do those interdigitations form, then you're stuck. You have to do it. If what you want to know is how do podocytes, once they've assembled in the kidney, coordinate to do filtration, then you may not need to model the generation of that structure. You may be able to model its effect. And in that case, the kind of thing we're doing here may be more appropriate. When you have very, very complex spatial architectures within an individual cell, there is no modeling package available today that is good. And that's not because people haven't tried, it's because fundamentally the physics of it and implementing the physics of it is very, very hard. People like Roger Kam, Karniadakis have done some very interesting modeling of this kind, but it's heavy. You're running supercomputers to simulate milliseconds and length scales of hundreds of nanometers. You're getting more in the world of molecular dynamics, macromolecular.
What if the kind of data was pointing to it being a chemical field that regulated it, and you knew what the bounds of that field were, and what kind of like, so your fiber length would be 10 nanometers under these conditions, but under these conditions it would be 100. Would that make it less computationally expensive to achieve?
Yeah, well, if there's anything you can do to do what's called homogenization, where what you care about is, say, the distribution of fiber lengths, or the density of fibers, or the average length of fibers, and you don't have to model the individual fibers, then things become much more tractable. So the moment you can represent something as a field, rather than having to represent each molecule individually, things become much, much easier.
Yeah.
And so I'd be happy to sit down with you and look at the problem. I'd be the first person to say that the kind of methods that we're doing here aren't going to be for everything. I think it's important sometimes to recognize what the limitations of methodologies are. And so I don't, but I think, I mean, I'm trying to understand, as I say, podocytes, of all the cells in the body, podocytes are the ones that intimidate me the most. So I think it's fascinating that you're working on them. I think they're incredibly important. How they work, there are certain specialized, I mean, neurons, of course, are the classic example of this, but there are certain specialized cell types in the body where you think, how did you get that? Podocytes would be one. The cells in the cornea are another. How do you build transparent cells that last?
I agree. I spent, I think, about 2 months last summer just trying to understand structurally how the podocytes were put together and held together and how they could move around to achieve their functions and stuff. But I do have like a good grasp of what's going on. I just don't know how to translate that into a model, as it were, yet. But
I'd love to sit with you and for an hour or two and learn about it and see whether I can help map some of that.
Yeah, that would be great.
But I don't mean to cut you off. You're addressing hard problems, and one thing you won't get from me. I may BS about some things, but you're not going to get any BS from me. If I think something's impossible or I don't know how to do it, you'll hear me say it. I'm never going to be ashamed of saying I don't know.
Yeah, and if you don't know how to do it, I certainly don't. So that would be a good starting point, at least.
But I can at least help explore it.
Yeah.
I'd be happy to.
Okay. Well, I'll email you. We'll set up a date, have a meeting and sort that out then.
That would be lovely.
Okay. Thank you very much. Good evening.
Thank you. Thank you. Anyone else have anything they want to discuss? It was a long day, but I hope it was productive for people. Okay. Do you mean now or at some future time?
Future time. It's just something for you, food for thought for some future philosophical discussion. It's a very interesting thing that I've become interested in. I would really love to talk to you more about it because it's a convergence of ideas that I actually learned from how cellular POTS models works. Mm-hmm. But it's an idea that's been brewing in something else, and I'm not that interested in how consciousness works and all that. So I don't want to scare you off with that. But there's this whole idea of the free energy principle and how cells are constantly, they are adaptive systems at every scale that allow the cell to move towards some parameter state that the cells have evolved to try to maintain. But I don't want to elaborate on it. It's a paper that when you have time, if you can look at, I would love to hear your reactions to it and discuss it.
That sounds like the kind of thing Itamar would also be interested in.
Yeah, so I've been throwing this idea around with other people, but they are not familiar with cellular POTS models and the way they work. And I think they've converged on a very similar set of ideas for completely different reasons. And so I think it's quite fascinating.
Okay. I'd be interested in hearing it. I tend to have rather strong feelings about anything where I hear optimum and evolution together, because evolution doesn't optimize anything except fitness, and fitness is an emergent property. It's not a property of the system itself. It's only
true.
A property of a system in context.
It's true. No, that's certainly true. Anyway, you can have a look at this paper. I completely agree with you, and then we can discuss why I think it draws me back to the principles by which CC3D or any cellular POTS model works, because I see some overlap, and I don't know whether there's a conceptual mistake in thinking there's an overlap or whether there's truly an overlap. That's sort of why I'm interested in thinking about it.
Well, so let me give you a bit of history, because this is something that Pedro brought up. Yeah, and I thought about giving some history of CompuCell methodology.
Very quickly.
We're out of time.
Ajay, are you comfortable posting whatever you message to James to everybody in the chat so we know what
sure
what you guys are talking about?
Sure.
I didn't know if it was
I'm sorry, I didn't realize I didn't
Yeah,
realize that that was only to me. I thought it was a general posting.
Yeah, I
in
figured
the
it was
paper.
supposed to just be a
People
quick
of
note to talk
interest.
about later, but I'm kind
Yeah,
of
that's
curious
what I
now.
intended because I know the time is up.
No, but I would say, so one of the things that we got a lot of flack for with the CompuCell, or the predecessors of CompuCell, the methodology, was that that the methods we're using. Let me see. There's the paper that's in the discussion. The methods we're using are doing something that looks like energy minimization. And actually what we're doing is solving forces. We're calculating local energy gradients and moving in the direction that reduces the energy, which is equivalent to saying that things move in a velocity proportional to force. So it's overdamped for force response. It does not minimize pattern energy as a whole, except under very specific circumstances.
Right.
And the physicists could not, and I started out as a physicist, the physicists could not get their heads around this. They kept saying, but this is not, the patterns aren't minimizing energy. Well, of course they're not minimizing energy. These are active patterns.
Correct.
These are living systems. Energy is continually being put into them. Now, locally, if I have something on a hill, it'll roll down the hill. So that's reducing the energy locally. But the hill itself could be moving up and raising the energy of the whole system. And so the problem is that the idea that there's a single functional that's being minimized is the problem. Physicists tend to think of evolution as optimization problem. If you could write a single functional that says, when you've optimized this, your fitness is best, then evolution would be extremely boring because you know what the answer was before you started. The problem is that when a system changes, its environment changes, its interaction changes, and therefore the things that you're competing with change continually.
Yeah.
And this is also happening within organisms. And the discipline of what's called active matter today in physics is asking the question, if your agents aren't just passive thermal agents, but are actually moving actively, then all sorts of things that are sort of physicists you never have equilibrium is death, right? So you never get equilibrium, you never get optimization, you don't get energy optimization. Now you get quasi-optimization because things have to be good enough. And TJ brought up bone refinement as an example. So we know that with the proper sets of stresses and strains on your bone spicules that get used a lot get thicker and are reinforced. Bone spicules that aren't used get thinner and eventually disappear. That's one of the reasons you get osteoporosis later on if you don't exercise right. Of course, if you give too strong a force, you break it, and then that's a different question. This happens, of course, with vasculature as well. Blood vessels that don't have a lot of flow shrink and eventually get pruned out, at least at the level of capillaries. Ones that are used a lot and have a high shear grow, get reinforced with parasites and become larger blood vessels. And so there's a continual flow of, there's a limit because you have a finite amount of energy to use, a finite amount of material, a finite amount of volume. But nevertheless, you have an exploration of space where if a region is hypoxic, it has neoangiogenesis. If a region has too many blood vessels, some of them disappear. But it's pretty fuzzy. It's not that you're going to have every point in your body have exactly the same oxygen tension. You can tolerate a lot of variability. And so I think this idea that things have to be pretty good, if things are terribly inefficient, if there's a way around it, then probably you'll find it. But you don't have to be perfect. Energy efficiency is not something that is
right. No, the idea I think that's being explored here is a much simpler one, and I think maybe the fact that I mentioned evolution was an optimization was a misleading representation of it. The core idea that I was really struck by, which I think is very interesting, is that in CompuCell3D, there's a very pragmatic thing that happens, that you set, say, the volume of a cell to be a certain thing, and that there is a cost to deviating from it, and the system tries to bring it back. And all I am emphasizing is that this is not just a thing that happens to be pragmatically useful for modeling biological systems. It is indeed what biological systems do.
Yes.
That is what
Well,
is so cool about it, I
Yes.
think.
Well, oh, absolutely. I mean,
That's all I'm
there I
saying.
agree completely.
That's all I'm
Homeostasis-
saying.
Homeostasis is
Correct.
absolutely
Correct. And
fundamental, and
understanding
it's
biological
mysterious.
systems.
How you generate homeostasis is incredibly difficult.
Correct. Because the cell doesn't know anything other than
Right.
it knows it can sense a deviation from something that it is set to, and it
Right.
has adaptive systems that bring
Right.
it back to that. That's
But
all.
it doesn't know what's going on 10 cells away.
No,
only knows it's
exactly.
lo-
Exactly.
I mean,
exactly.
if you try to build a building from the inside with only
Yes.
local information, if you
Yes.
can't see, we're so dependent on being able to
Exactly.
see distance.
Exactly.
Trying to do things with only local information is really hard.
Yeah.
The principles of organization are totally different. Self-construction and self-maintenance are totally
Yeah.
different. So I agree with you completely that that's
Yeah, so
important.
this is why this paper is interesting, because it comes to that same idea from a completely different origin. And I think it will be interesting for you to read about it, because you came to it from a very different direction, I think. But it's sort of a convergence, which is very interesting.
But I agree with you completely. And if you talk to the top, I learned, I mean, the people who helped me appreciate homeostasis as a problem were the toxicologists.
Yeah.
Because physicists, again, if you're not doing something to a system, it doesn't do anything, right?
Yes.
It's Newton's law. If you don't do
Yes.
anything, it stays there.
Yes.
Biology, that's not true.
Exactly. It persists
so-
not because it's inert,
Right.
but it's because it's constantly
Right.
bringing
Right.
itself back to
Exactly.
that state.
Which is why in my diagram that I use all the time of the embryo, the mouse, and the dying mouse,
Yes.
I say, why is the mouse the same in the two pictures?
Yes.
I say that's the hardest problem of all. How do you
Correct.
keep the mouse the same?
Correct. And the thing is that it's doing that at every scale within the living system. That's the cool thing.
Absolutely. Absolutely.
Anyway, I also have to run, but I'm
No,
glad I
that's
got a
great.
little
Well,
chance
thank
to
you. I'm
introduce
glad we
this to
got
you.
to chat. And I know Idemar is interested in these problems too, so I'm glad we got to have a little chat about that. Thank you. That's great.
Okay. I'm enjoying this, even though I'm doing this for the nth time. I'm constantly learning. I'm still so much to learn, and it's just great being here.
Okay, well, thank you for joining us. I appreciate it.